Chandler Connor.
Nathan Marin.
I'm Brian Darst.
And I'm Matthew Stafford, and we're presenting DNA replication in E. coli.
This is Ori C, the origin of DNA replication in E. coli. This region of the bacterial chromosome contains cis-acting elements that are critical for the recognition of proteins involved in the replication process. Reading left to right, the top strand is 5' to 3', and the bottom strand is 3' to 5'. The 10 DNA A binding sites are 9 base pairs long. The area in which they are found is also known as the recognition element. The DNA unwinding element, or DUE, is a tandem array of 13 base pairs conserved sequences. A protein called DNA A recognizes the DNA A binding sites, altering the shape of the DNA and causing the duplex strand of DNA to melt the DUE. The separation of the two template strands forms a replication fork, marking the site of synthesis of the daughter DNA strands.
Next, DNA B, a hexameric helicase, is recruited to the replication fork with the help of DNA C. The helicase is added to both template strands, allowing replication to occur bidirectionally. The helicase action of DNA B further unwinds the DNA downstream, resulting in a replication bubble. DNA C then leaves the region after DNA B is bound to the single strand. DNA B also promotes binding of DNA G, a primase. There are actually 3 primases bound per helicase in E. coli, but here we will just show one. Together, these factors form the primosome.
The primase DNA G next to the DNA B helicase attaches an RNA primer to the parent DNA strand. The clamp loader then recognizes the replication fork. The clamp loader is composed of 3 tau subunits, which interacts with the core polymerase, the beta clamps, and the template strand. Delta subunits open the beta clamp, while the delta prime subunit regulates the delta subunit. The chi subunit binds single-stranded binding protein to the exposed single strand. Psi subunit connects chi to the clamp loader. The clamp loader then recruits a beta dimer to the prime template to form beta clamps. One clamp binds to one strand, while the other beta clamp binds to the primer. Binding of the beta clamp causes it to have a high affinity to core polymerases. The tau subunits of the clamp loader bind core polymerases to the beta clamp. One polymerase binds to the beta clamp on the leading strand, while another one binds to the beta clamp on the lagging strand. Everything is now ready for DNA replication.
During replication, the parent DNA is constantly being unwound. On the leading strand, the bottom strand, the daughter DNA is continuously synthesized 5' to 3' by the polymerase as it progresses toward the replication fork. The lagging strand runs from 5' to 3'. Since DNA can only be synthesized 5' to 3', a structure called the trombone loop is formed. The second core polymerase binds at the clamp. Single-stranded binding proteins, shown in black, bind to the exposed single strand in the trombone loop. to protect the strand from nucleases and prevent the strands from premature annealing. The second core polymerases synthesizes a new daughter strand starting at the primer as the leading strand continues to be synthesized. When the second core polymerases reaches another primer, the polymerase and clamp dissociate from the DNA. The polymerase is still attached to the tau subunit. The fragment of DNA synthesized between the primers is known as an Okazaki fragment. The fragments are ligated together later to form a continuous strand of DNA. While the second core polymerase is synthesizing the daughter strand, single-stranded binding proteins dissociate and the clamp loader brings in a new beta clamp at the new primator, located upstream on the parent DNA. A tau arm brings a third polymerase that binds to the new clamp and synthesis of the new Okazaki fragment begins.